This is an A-level biology presentation looking at HIV and the ELISA test. In this particular presentation, we're going to look at the structure of HIV, how HIV and AIDS are connected, and how HIV actually is able to replicate and infect a host. We're also going to look at the processes involved in the ELISA test. To get started, there are 6 different variables that need to be considered when rolling out a vaccination program. Look at each of the 6 pictures, try and work out what they might represent in terms of how it could be effect on a vaccination program and how successful it is. And when you're ready, you can restart the presentation. So pause the video now. There are 6 different pictures, and if you've gone through some of the other presentations on vaccines, you will have seen this already. But what we've got is it needs to have few side effects, it needs to be economically viable as a vaccine. We need to make sure we can produce large enough quantities, we can store large enough quantities, we can transport large enough quantities, and when we get it to where it needs to go, we've actually got a method of administering the vaccine to individuals as well. HIV is an RNA virus and is classified as a retrovirus because it takes its RNA and it uses an enzyme called reverse transcriptase and it converts it into DNA. There are treatments available. There are different types of antiviral drugs that will slow the spread and production of HIV, but currently there is no cure. It is transmitted through sexual intercourse, but there are other methods of transfer, for example, through blood and breast milk. It is not transmittable through sweat, urine, or saliva. You cannot kiss someone and pick up HIV. It has to be through blood. Those are the most common way. Often it's found commonplace between those with drug addiction because of the sharing of hypodermic needles, although many, certainly first world countries now, have schemes in place where if you are a drug user, you can go and get replacement needles to stop the spread of HIV. And of course, as it is a virus, it can only replicate inside a host cell. It is only active when it gets inside a host. So this is the structure of HIV. If you have not taken the time to memorize this, there's the first time you're going through this presentation, I'd suggest you go straight to the answers and memorize each of the different components and then go back in a little bit of time and test yourself and see if you can still identify them correctly. Remember, don't learn the number sequence for to fill in the blank spaces. You need to be able to draw and label that diagram from memory. It's not just enough that you can put in the sequence of numbers on the right hand side of the task. Easiest way to check, get a blank piece of paper and from memory draw and label and see if you can come up with exactly what's on the screen now. If you don't, then I suggest you go and spend the time learning it before you progress any further through this presentation. Pause the presentation now, and when you're ready you can restart to go through the answers. Answers are as follows. Number 1 is what we call the matrix. Number 2 is what you call the capsule. Number 3 is the actual RNA, the genetic material of the HIV virus. Number 4 is what we've mentioned earlier, is the reverse transcriptase enzyme, and its function is to take the RNA strand found inside HIV and when it infects a host cell to turn that RNA into DNA. We also have what's called the lipid envelope, which is the outside layer, and then finally we have what are called attachment proteins, and the attachment proteins are the way in which the HIV basically binds to specific types of cells. In front of you, you have the process of HIV replication. Each picture represents a particular part of the stages of replication, from how it first gets into the blood all the way through how HIV is then released out from a particular infected cell. All you have to do is go through each of the pictures, describe what's happening in the picture, and then when you're ready we can go through each of the different slides individually and give you a kind of overview of the process. So pause the presentation now, try and work out all 9 images, and when you're ready you can restart and we'll go through them. So in the first picture, what we've shown is clearly a hypodermic needle. So for some reason a hypodermic needle has transferred the HIV virus into the bloodstream, and we got to talk about the bloodstream so we're clear, we know how HIV is transmitted. In the second slide, what we've got is the HIV virus has now bound to what we call a CD4, which is a type of protein found on the surface of T helper cells. If you don't know what a T helper cell is, please go back and look at the other presentations on the immune system, specifically the T cell one and the B cell one would probably help as well. Once we have the HIV attached to the protein on the surface of the helper T cell, the inside layer, which is known as the capsid, releases and moves towards the cell surface membrane. At the cell surface membrane, the capsid stays there, but into the T helper cell we release 2 things, which is the RNA from the actual virus and of course the reverse transcriptase. Once inside, the reverse transcriptase converts the RNA from the HIV into the DNA strand. That DNA strand, which is the pale blue color, is then inserted into the nucleus and into the DNA of the host. Once it's in the DNA of the host, by transcription and translation, that host, just as it normally would, starts to produce new structures and new proteins and new polypeptides. But because it now contains the DNA that corresponds to the HIV, the cell starts to produce all the components that are needed to build another HIV cell. And it actually produces them in very large amounts, and it produces them very, very quickly. The outer layer of the HIV comes from the process involved on the HIV when it leaves the cell. It actually takes away part of the cell membrane from the host cell that it's been infected, and that's how we get the outer layer around the HIV, and then that HIV virus is then able to go off and infect another cell. HIV and AIDS are both caused by the same virus. What happens is that HIV targets T helper cells, and when it is inactive and it's not causing any particular problem to the person's immune system, it is referred to as HIV. However, when that HIV starts to destroy enough of the helper T cells that it impacts on the person's immune system, then we start to refer to the particular disease as AIDS. When a person has AIDS, they are more likely to get a secondary infection. They are more likely to pick up things like pneumonia, and as a consequence the body's not able to fight those infections off because of the weakened immune system. So people don't die from HIV and AIDS directly. The HIV and then AIDS will basically destroy their immune system, and without the immune system, things that they would normally be able to deal with, as I said, like pneumonia, then become deadly, and even things like flu. Second part of this presentation is looking at what's called an ELISA test. An ELISA test is a very, very sensitive test that lets us detect trace amounts of protein. And we use it to check for a number of diseases, including HIV, but we can also use it for a number of other purposes as well, for example, drug testing and so forth. What you have now is the 8 main stages involved in the ELISA test. You have to be able to describe how to do an ELISA test. You'll also have to be able to describe how to interpret results from an ELISA test. At this point, pause the presentation, work out what each of the pictures tells you, and when you're ready you can restart the presentation to work out what happens in an ELISA test. So these are the main stages in an ELISA test. Step 1, we have to get a sample that we are trying to test to see if it contains a certain protein, substance or pathogen. And we have to get it so that it is fixed to a slide. It cannot dislodge or be removed from the slide. Then what we do is, just to make sure we don't have anything on there that shouldn't be on there, is we wash and remove any part of any antigens or any pathogens or any substance that is not fixed to the slide. Then what we do is we introduce our first antibody. By introducing our first antibody, that antibody will attach to the antigens of the particular substance we are testing for, or particular type of pathogen we're testing for, if it is present. We do have to wait a period of time for that to occur. After a period of time, any antibodies that can attach to the antigens that are fixed onto the slide will do so. We then wash the sample again so that any antigen that is just free floating on the surface is removed. And we know that any antigens that are left are only there because they have attached to any antibodies left have attached to the antigen of the cell, pathogen, or substance that we are trying to identify. We then add in a second antibody, and that second antibody contains an enzyme. That antibody is complementary to the first antibody. Because the first antibody is attached to the particular antigen that we're testing for, the second antibody attaches to the first antibody, which is attached to the antigen and the pathogen, which is fixed to the slide. As a consequence, the enzyme that's attached to the second antibody, we can then use that as a substrate being converted from one type of substrate to another type of substrate, resulting in a color change. If the particular pathogen that we're testing for, substance that we're testing for, is present, we will see a color change in the substrate. We can even then start to relate the intensity of color change with how much enzyme is there, hence how much of antibody 2 is there, which means how much antibody 1 is there, which means how much antigen is there, and it gives us an indication of the concentration of the pathogen or particular substance that we're testing for. So there's a relationship between the color intensity and the amount of antigen that is present. And these are the stages and main stages involved in the ELISA test. You have to be able to describe that process and the interpretation of the results in terms of intensity of color. It is assumed at A level that you do remember from GCSE what an antibiotic is. Should you need to, there is a presentation on antibiotics and painkillers that you may want to check out. An antibiotic is a particular type of medicine that is used to treat bacterial infections. It will kill bacteria. It does not affect viruses. One of the most common questions in A level is, why does it not affect viruses? Well, first off, you need to know how an antibiotic works. An antibiotic works by basically weakening the cell wall of the bacteria. By weakening the cell wall of the bacteria, as we have osmosis and water moves into the cell, we basically have a cell wall that cannot support the cell bacteria, and as a consequence, the bacteria lyses or bursts open. The problem we've got is that works with bacteria, but it doesn't work with viruses. Now, the reason it doesn't work on viruses is a little more complicated than you might think. Yes, viruses are found inside a host cell. Because they're found inside a host cell, the chemical antibiotic floating through the blood does not reach them. It goes a step further than that. Viruses don't have a cell wall made of murine, which is the substance that is targeted and basically compromised by an antibiotic. They have a protein coat around their outside surface, so antibiotics don't affect them for that reason too. What we also have is that a virus has no metabolic pathways. There's nothing to interrupt, there's nothing to break into, there's no osmosis moving in and out, there's nothing inside that cell happening. That cell injects its DNA into the host cell, and that's when replication occurs. And just to finish off, last thing I'd like you to do is you should have gone through and memorized the process of how HIV replicates and the process of carrying out an ELISA test. all you have to do is, from memory, is produce 2 flow diagrams, one for HIV replication, one for an ELISA test. However, you're not allowed to use more than 50 words to cover both of those flow charts.